Hello, my name is Ren Li, a PhD student from the Aphasia Research Laboratory at Boston University. Welcome to my presentation of our recent work on predicting typicality effect from brain activation patterns in healthy adults and individuals with aphasia. The neurorepresentation of semantic processing has been studied for many years in both healthy individuals and people with aphasia. For general semantic processing, previous work in healthy adults has suggested a left lateralized neural network, including temporal, frontal, and prefrontal regions. Other works have proposed the recruitment of anterior temporal lobe and other regions for semantic control. In people with aphasia, the neural network for general semantic processing is much more distributed. Another line of research in neurorepresentation of semantics has focused on a more granular level of processing, which is category-specific. Previous work has suggested different accounts for category-specific processing in healthy adults, but evidence in people with aphasia is still limited. Another dimension of semantic processing is semantic typicality, which means more typical members of a semantic category share more semantic features in common with other members of that category, while less typical members share fewer common features with other members. Evidence of the neurorepresentation of semantic typicality has mainly been derived from healthy adults, suggesting recruitment of the visual and temporal regions. It is also reasonable to assume that people with aphasia may expect real-world object categories to have a strong prototype-dominated cortical representation, which might be different than healthy adults because the language network is damaged after stroke. So the current study aims to use a more sensitive approach to detect which brain regions show neural encoding of semantic typicality in both healthy adults and individuals with aphasia. There were 21 individuals with chronic stroke and 18 healthy adults included in the current study. Prior to scanning, all the stroke patients were administered a battery of standardized language assessments. And in the scanner, the participants completed a semantic feature verification task, which included typical and atypical exemplars of different semantic categories. Behavior performance of the fMRI task showed a typicality effect, which means both groups of participants responded to more typical items faster and more accurately than atypical items. The fMRI univariate analysis examined neural activity between these two conditions of stimuli. Results here are uncorrected because we did not find any significant clusters after correcting for multiple comparisons. For the contrast, typical versus atypical, we found neural clusters in the right supramarginal gyrus and right middle cingulate. For the contrast, atypical versus typical, we found a cluster in the left middle occipital gyrus. Our searchlight multivoxel pattern analysis showed two clusters with significant above-chance classification accuracy after correcting for multiple comparisons. One was in the left middle occipital gyrus extending into the left lingual gyrus, and the other one was in the right calcarine cortex extending into the right superior occipital gyrus. We repeated the same analyses in stroke patients. There was no significance in the fMRI univariate analysis with or without correcting for multiple comparisons. The Searchlight MVPA showed two clusters of voxels with above-chance classification accuracy. One was in the right rolandic operculum, and the other one was in the left fusiform gyrus. However, no significant results were found after correcting for multiple comparisons. We additionally examined the correlation between the neurorepresentation of semantic typicality and behavioral performance in individuals with aphasia. We found that accurate response in the fMRI task significantly predicted the neuroclassification accuracy in the left middle occipital gyrus in patients with anomic aphasia, rather than in those with Broca's aphasia. In summary, our main findings suggest a similar behavioral typicality effect in both healthy adults and people with aphasia. The semantic typicality was represented in the visual cortex in healthy adults, which was different in people with aphasia. Although the searchlight analysis did not show any significant results, there was still a strong association between the visual representation of semantic typicality and accurate response to typical versus atypical stimuli in patients with less severe aphasia, suggesting a less automatic neuroprocessing of semantic typicality in people with aphasia compared to healthy adults. Thank you very much for your interest, and I hope you enjoyed this presentation. If you have any questions, please do not hesitate to contact us via email.